with talking about nervous tissue physiology, which is the electrical activity stuff. Hopefully we'll finish that up here because we're pretty much at the end of that. And then we'll start talking about the nervous system chapter, which will get us kind of caught up with where the lab material is. So let me go back to the presentation. I was using presentation stuff to talk about stuff like, okay, good. And I saw on Casey's notes that I talked about propagation, so I must have shown you this picture, right? And then I showed you this, or a video, maybe not this one, but a video about propagation. Actually, the one I showed you, was it the Pearson video? Okay. So I didn't put the Pearson video in the presentation, which I think I actually did right after class for you guys. And this is just another example of propagation, which actually offhand, I don't remember what this goes into exactly, but it's another video sort of covering the same material. And then you had written down in your notes graded potentials, but not really anything after that. So did I not really? Sorry? We did go through all these. So we looked at, okay, good. So we looked at this. Graded potentials are different sizes based on the size of the stimulus. They can be depolarizing or they can be hyperpolarizing. Depolarizing ones are called, at a synapse, a depolarizing graded potential is called an excitatory postsynaptic potential. A hyperpolarizing graded potential at a synapse is called an inhibitory postsynaptic potential. And those two events can add up, summate at the target neuron and hopefully get to the point of hitting threshold and causing the action potential at the axon hillock. Okay. So the last part of this is dealing with the synapse. So we hadn't gotten to any of this yet, right? Okay. So this is the picture of, whoops. Oh, first off, I keep forgetting it goes in here. There are actually two types of synapses. There are chemical synapses and electrical synapses. We're just going to talk about chemical synapses here. And that's where neurotransmitters released from one cell onto the next cell. Electrical synapses are where there's a physical connection between two cells, and ion channels go through the membrane of the two cells, and the ion channel is actually a continuous connection, so that if sodium gets into one cell to depolarize it, that sodium will automatically cross over the connected membranes and depolarize the neighboring cell. The only place where that's relevant in A&P is in cardiac muscle tissue. It exists in other places and in some places in the nervous system, but we're not studying the nervous system to the level that we would care about electrical synapses there. They're used in special situations that aren't relevant to what we talk about in this class, but they do exist. And so I'm just mentioning that so that when we get to cardiac muscle tissue in A&P2, I'll just come back and remind you of this stuff. What I put in here, helping to define the chemical synapse, just tells you what the elements of it are. And an important part to consider is that the chemical signal will cause changes in the target cell, the postsynaptic cell, as long as it's there. So we have to remove the neurotransmitter from the synapse. And there are basically three ways that can happen. Either it can just diffuse away, which is not a great way to do it because that chemical then could go somewhere else and have an effect. It's not very likely, but it is possible. It's not the most controlled way of taking care of it. Secondly, an enzyme could break that molecule down, and when it's broken down, it's no longer a neurotransmitter. And then the other possibility is that cells in the neighborhood, usually the presynaptic neuron or possibly an associated glial cell, can reuptake the molecule, which basically means to pump it back into the cell or pump it into the cell to clear it away. And so those are things that we want to think about. In the book, I have this same kind of information. I think it's just a little bit more extensive, talking about the parts of what makes up a synapse. So this is a picture just showing the main parts of the synapse. Since I showed you that picture of the propagation stuff, I must have been talking about the other textbook that has those pictures in it, right?
The biology.
Right. So the biology textbook has this picture in it essentially, but it also has two little insets, which in retrospect I'm going to swap out and put in place of this, because one of those insets actually shows the 3 different ways that you can clear neurotransmitters out of the synapse. The other inset, I think, is basically what we've looked at several times, which is neurotransmitters binding to the receptor, which we have other pictures of ligand-gated ion channels, which is what those are. But I like that other inset, so I'll probably include that in when I finally get around to doing the recording, which I'm going to start tomorrow because I finished all my grading this morning. Yay! Okay, but that's neither here nor there. But we talked about the neuromuscular junction and the muscle system, and that's just a specific example of a synapse. So it's essentially the same thing we see here. The presynaptic neuron, its axon ends what we call the axon terminal, and there's an enlargement at the end, which is not labeled here, but called the synaptic end bulb, which contains vesicles filled with neurotransmitter. And when the action potential gets to the end here, the depolarizing current in the membrane will activate these channels here, which are not labeled in the picture now that I notice it, but they are voltage-gated calcium channels. And when calcium enters in through these channels, it binds to proteins on the surface of these vesicles, which causes the vesicles to merge with the cell membrane and release their contents. Now, calcium is a positively charged ion and would therefore also depolarize things at that point. But the depolarization is not important. At this point, we're just about releasing the chemicals. And calcium is an ion that is often used to help proteins do their jobs. Where's another example that we've seen calcium do help a protein do its job? What's really the only other time we've talked about calcium in this question? Oh, good point. We've talked about it more than once. Muscle contraction, yes. So in bones, yes, that's where we store calcium, but it's not about interacting with proteins. But in muscles, calcium binds to troponin C, troponin moves tropomyosin out of the way so that myosin and actin can interact. That's what I was talking about. I completely forgot about, yeah, we did talk about calcium in bones. So calcium tends to be involved in attaching to proteins and making proteins change their behavior, I should say. So that's what calcium does here. It attaches to proteins on the outside surface of the vesicles, which makes the vesicle dock to or merge with the cell membrane and therefore release their contents. And then their contents, the neurotransmitters will diffuse across the synaptic cleft, which is a very, very short distance measured in nanometers, and bind to their receptors on the opposite side. The picture from the muscle chapter was essentially the same picture, but instead of a yellow thing down here labeled postsynaptic neuron, it was red and it was labeled muscle cell. Okay. But it's just the target. Here, since we're talking about the nervous system, we're talking about synapses between neurons. So we have the presynaptic and the postsynaptic neuron. When these ion channels open up and sodium rushes in, that's going to depolarize the target cell, and that's going to be the postsynaptic potential. And since I just said it depolarizes, that would be an excitatory postsynaptic potential. It's possible that other things could be, other types of receptors could be there. So I want to talk about receptors a little bit more and the various types of neurotransmitters out there. First off, receptors fall into two basic categories. The first one, which is actually what we've been talking about for a while already, is called an ionotropic receptor. The ending there, tropic, kind of means it's related to the word movement. How exactly, I don't know, but ionotropic basically means receptors that move ions. That's a very crude definition, but let's work with it. And this is a ligand-gated ion channel, which we've already talked about plenty. In this picture, for some weird reason, it shows a third molecule attached to the outside surface. But really, it should be two, and that would be the acetylcholine receptor that we already talked about at the neuromuscular junction, and it's been the example ligand-gated ion channel, as we've talked about it several times so far. But the molecule binds to the receptor and it opens up so the receptor will allow ions to cross through the membrane. The other type is called a metabotropic receptor, which, if my definition of ionotropic was crude, trying to define this one likewise is really crude. But basically what it means is that it causes metabolic changes in the cell. Ionotropic directly opens ion channels. Metabotropic receptors cause chemical changes or metabolic changes in the cell. So the receptor is a protein that spans the cell membrane, just called the receptor protein. And on its inner surface, it's attached to a protein that's called a G protein. And the G in G protein stands for GTPase. Now you might have picked up on the occasional reference to an ATPase, which is an enzyme that hydrolyzes ATP to get energy. A GTPase is essentially the same thing, except that it primarily will hydrolyze GTP, which is just a variation of what ATP is. It's a different version of the same kind of molecule. When GTP binds to the G protein and gets hydrolyzed, then the G protein is activated and will bind to another protein, which is called an effector protein. The effector protein is a specific enzyme that is what causes the metabolic changes. And this particular example here uses ATP to make another type of molecule. So ATP is a substrate, and these little dots here represent the molecule that's made from it. That's what's called a second messenger. And these little lines are representing the second messenger traveling within the cell and doing things. And the thing that it's doing is it's opening an ion channel. So here, ultimately, we have receptors that open ion channels. Ionotropic receptors are directly opening an ion channel. They are ion channels. Metabotropic receptors indirectly open ion channels. So the receptor binds to something, and it causes changes inside the cell that will open the ion channel. Now, in this picture, I kind of wish there were multiple ion channels represented here, because that's in fact the case. One receptor can open a number of ion channels, the metabotropic type. Whereas for an ionotropic receptor, it is one ion channel. It can't open up more ion channels. More neurotransmitters can open up other ion channels, but you need more neurotransmitter to open more ion channels when they're this type of ion channel. But this one receptor that's bound to 2 or 3 whatever molecules can open multiple ion channels. So there's a trade-off here. The positive effect of metabotropic receptors is that you can actually open multiple ion channels at once. The downside of metabotropic receptors is that you have to have a few different reactions take place. The metabolism has to change inside the cell, which can take a little bit of time. It's an amount of time that's measured in maybe hundreds of milliseconds. It might be 100, 150 milliseconds worth of delay between binding to the receptor and opening the ion channels, which isn't a huge delay. Our conscious perception can't really tell the difference between instantaneous versus 150 milliseconds. Just too quick of a time frame for us to really notice consciously, but it is a delay. The direct action of ionotropic receptors is instantaneous. As soon as this molecule binds to the receptor, to the ion channel opens up. It's an instantaneous activity. So you find these type of receptors in different places for various reasons, but it's going to have something to do with the trade-off between instant results versus possibly widespread results, meaning multiple ion channels. For instance, when you want to activate a muscle, like you touch a hot stove and you want to pull your hand back, you don't want to wait 150 milliseconds for that to happen because you will get tissue damage as you burn your fingers. So muscle contraction is always controlled by ligand-gated ion channels. Neuromuscular junctions have this type of receptor there, so that there's an instantaneous depolarization and the fastest possible response to withdraw the hand or whatever movement of the muscle you need to perform. Metabotropic receptors are found in other places, keeping with the controlling muscles. Controlling smooth muscles or cardiac muscle isn't as time sensitive, so this type of receptor is fine. For example, cardiac muscle actually contracts on its own. It doesn't need any outside influence to tell it to contract. And so what our nervous system does to cardiac muscle is speed up the rate of contraction or slow down the rate of contraction. So if you want your heart rate to speed up, this kind of receptor is present in the wall of the heart, causing changes. And the hundreds of milliseconds worth of delay between binding and something happening isn't as important as in skeletal muscle that achieve instantaneous reactions. So there's going to be trade-off between these two types of receptors, and they're going to be used in different situations for whichever type is best suited to what's going on there. Now, I want to talk about the different types of neurotransmitters, and I have this table here. Now, there's a table in the textbook, and actually this is that table, I just improved on it. The table in textbook, if you look at it, it's actually kind of rotated 90 degrees. So the first column in the textbook version of this table is the top of the table. So it says neurotransmitter receptors, method elimination, postsynaptic effect down this side, and lists things out that way. In retrospect, it was not the best way to organize the information. So I rotated around so that I could put more correct information in. So on this side, we have the name of a number of different neurotransmitters that I want to talk about. Acetylcholine is one that we've already talked about, and it is abbreviated capital A, capital C, lowercase h. I would strongly suggest that you try to use the abbreviation more than trying to spell the whole thing out. Students will often misspell acetylcholine. The most common misspelling I see is that they leave the L after the Y out. So it's acetylcholine or something like that, which is a misspelling. On autocorrected questions, if you misspell a word and it doesn't match what the computer's looking for, it's wrong. Might be obvious that that's what you meant, but doesn't fit what the computer's looking for. And I'm not going to tell a computer, I want acetylcholine or something close to that. You have to be sure that you spell things correctly when you put them into autocorrected fill-in-the-blank questions. So I always put the abbreviation as an alternate possible entry. So please it's a lot easier to spell ACH than to spell acetylcholine. I strongly suggest you stick with the abbreviation there. That's essentially true for all the rest, if you want to do that. Now, acetylcholine binds to two different types of receptors, what are called nicotinic receptors or muscarinic receptors. Now, the types of receptors here, the names that we have for them, are based on drugs that bind to them also. Now, the idea behind drugs, as far as we're concerned to describe these things, is that they're tools that can be used in the laboratory. So if we're studying the effects of acetylcholine in the body, and we want very detailed information about what it does, just using acetylcholine in the laboratory to activate receptors is actually not the best way to do it. Instead, we have these drugs that we can differentiate between the types of receptors that acetylcholine binds to. So obviously, the nicotinic receptors are activated by nicotine. That's what they're named for. Don't get caught up right now in the fact that it is nicotine. You've probably heard of that. You know where it comes from, et cetera, et cetera. That's not important at this point. It just happens to be a molecule that we can artificially use to stimulate these receptors. So think about it in a laboratory setting rather than in a substance abuse setting like smoking. So we can actually manipulate that type of receptor and leave the other types of receptors alone. Okay? And likewise, the other type of receptor is activated by a drug called muscarinic, hence the name. And if we wanted to say this receptor not affect nicotinic receptors, then we could apply that drug instead of using acetylcholine itself. That's all it is. One way to think about what receptors and their ligands do is to think of them as keys and locks. So imagine I have 3 keys, okay, and 2 locks to think about. Now I used to actually have these keys. We used to work this way in our department. I'd have a key that I considered my office key, and I could open my office door with it. But it was actually a submaster key for the department, and so I could use my office key to open the classroom doors or the AV room doors. Okay. It would open any of the doors in the department that I was supposed to be able to open. I think like the dark room door didn't work with the master or something like that for a weird reason. But we don't even use those keys anymore. So we rekey everything. But the story still works. Okay. So I have the submaster that can open really any door in the department. And then I had 2 other keys, which all faculty get. They get a classroom door key and they get an AV room door key. So I could use my classroom key to open this door too, but I could not use my classroom key to open the AV room door. I could use the AV room door key to open the AV room door, obviously, but I couldn't use the AV room door key to open up the classroom door. You see how I have these different types of locks and keys, and they each have their specific locks that they work. Everybody with me on this? Okay. So in this story, my office key, which was really a submaster key, was acetylcholine. It could open up multiple types of doors. And then the classroom key, let's say that would be nicotine. It can only open the classroom door like nicotine can only activate the nicotinic receptor. And then the AV room key was like muscarinic, it can only open the AV room door. Okay. So I could use one key to open all of the doors, but I also had these other keys to open specific doors as needed. And that's sort of like this. We actually could use acetylcholine on either of these, or we could use nicotine just on the nicotinic, or muscarine just on the muscarinic. Again, it's really an artificial setup in the laboratory to study what's happening here. Now, my key and lock example there kind of illustrates what I've gotten written up here, but it is a little bit more complex than that. There are actually subtypes of muscarinic receptors, but that level of detail is not necessary for this class, just the idea that there are different types of receptors, 2 different classes. There are not subtypes of nicotinic receptors. Okay? And nicotinic receptors are the receptors we know about. So anything we've already talked about that acetylcholine binds to is a nicotinic receptor, which means nicotinic receptors are ligand-gated ion channels or ionotropic receptors, however you want to describe it. Muscarinic receptors, which we haven't talked about yet, happen to be the metabotropic style receptor. And I did give you the example of controlling cardiac muscle tissue, and acetylcholine, while it is active at the neuromuscular junction for skeletal muscle, is also active in controlling cardiac muscle. And so muscarinic receptors do that. If you were to put acetylcholine onto cardiac muscle tissue, it would bind to the muscarinic receptor, and it would cause the muscle tissue to slow down its rate of contraction. Okay? So that's, chemically speaking, what our body does to decrease heart rate. Now, I also want to put in here just a list of how we eliminate things, which is not vitally important, but I wanted you to see sort of the range of different things that happen. So for acetylcholine, it's degraded by enzymes, or an enzyme, and that enzyme is called acetylcholinesterase. If you have to abbreviate that, it's ACHE. The A, the C, and the E are all capital letters, and the H is lowercase letters. So it's acetylcholine, and then the E for esterase, which is the type of enzyme it is. Now over here under postsynaptic effect, I just tell you what that neurotransmitter is capable of doing. When acetylcholine binds to the nicotinic receptor, it causes depolarization through direct effect, which means the ionotropic receptor directly opens an ion channel. When acetylcholine binds to the muscarinic receptors, they can cause either depolarization or hyperpolarization, depending on the exact subtype of muscarinic receptor, and all of those are indirectly going to open ion channels. Those are metabotropic receptors. So this is the kind of information I'm providing for these different neurotransmitters. Now you'll also notice on the table, and this is something that was a shortcoming of the version of the table that's actually in the book. I have them sort of grouped out in shaded regions. Acetylcholine is kind of its own thing. And then these next three are one type, and these next three are another type. Acetylcholine is what we call a cholinergic neurotransmitter. Nicotinic and muscarinic receptors could also be called cholinergic receptors. Cholin in cholinergic is just from the word choline. And then ergic, the root of that is the root of the word energy. So it means the neurotransmitter system that's based on the activity of acetylcholine. And there's not another neurotransmitter that falls into that class, so it's cholinergic by itself. Now, in the next region here, we have three different neurotransmitters, and they're grouped together in sort of the unshaded part of the table because they are all amino acids. Now, acetylcholine is kind of a unique system, so we just call it the cholinergic system. These are all amino acids, but glutamate, glycine, and GABA work independently, so we talk about the glutamatergic or the glycinergic or the GABAergic systems. That's why I have sort of three different things. Each of these 7 things here can be referred to as something ergic systems. The shaded region that acetylcholine is in is to say that's different from the rest. And then the unshaded part that glutamate, glycine, and GABA are in is because all of those are amino acids. And then when I get down to this bottom part, these 3 are grouped together in a shaded region because they all share another common characteristic. So glutamate, which is abbreviated just GLU, and usually when you're abbreviating amino acids, you just abbreviate them in lowercase letters with a 3-letter code. Usually. Now, the ones that we abbreviate like that, which includes glutamate and glycine, are also amino acids that are commonly used to make proteins in our body. Okay? So amino acids do more than just act as subunits of proteins, and here they're used as signaling molecules. Glutamate binds to glutamate receptors, and we just refer to them by the abbreviation GLU capital R. Capital R stands for receptor. There are multiple versions of this, but I don't care to break down the different subtypes there, kind of like with muscarinic. For really all amino acid neurotransmitters, they're cleared away from the synapse by reuptake, either by neurons themselves or glia around the synapse. All glutamate receptors cause depolarization. Okay? And depending on the subtype, some of them are ion channels themselves, and some of them are metabotropic receptors. So it works slightly differently depending on the particular version, but they all cause depolarization. Sometimes we will refer to glutamate as an excitatory amino acid. Okay. You might hear that reference. It's actually a little bit imprecise to say that. The amino acid or the neurotransmitter, whatever type it is, isn't necessarily excitatory or inhibitory itself. What it is that the receptors are the ones that cause depolarization or hyperpolarization. Since all glutamate receptors cause depolarization, the neurotransmitter that binds to them can be considered excitatory because the end result is an excitatory postsynaptic potential. Glycine has, as far as I know, only one type of receptor. So it doesn't say multiple here because there aren't multiple types, but there's the glycine receptor. Again, reuptake through neurons and glia. The glycine receptor always causes hyperpolarization through a direct effect. It is a chloride ion channel. So, actually, if you put the nicotinic receptor and the cholinergic receptor side by side, you have two ligand-gated ion channels, one opened by acetylcholine, the other opened by glycine. The nicotinic receptor allows positive ions to cross the membrane, and the glycinergic receptor allows negative ions to cross the membrane. Now, positive ions could go either way, whether we're talking about sodium, calcium, or potassium. Chloride's only going to enter into the cell because that's the way its concentration gradient works. Okay. But when you allow negatively charged ions into the cell, it's going to cause hyperpolarization. Then we have GABA. Now, GABA is not like the other two amino acids here. It is an amino acid, but it's not actually the type of amino acids that's used to make proteins. And GABA is an abbreviation. It's an abbreviation for gamma amino butyric acid. Don't bother with the full name there. Don't ever use that. We always just call it GABA. And there are multiple GABA receptors, again, like the glutamate receptors, but we're not going to break them down into subtypes. All GABA receptors cause hyperpolarization, some of them through direct effect, some of them through indirect effect. I'm actually not an expert on GABA receptors, so I'm not entirely sure how all of them work, but they're going to open up ion channels that are either going to allow chloride in or potassium out. Both of those things cause depolarization. I just can't remember how common one versus the other is. It depends on the particular receptor. So again, those are all amino acids. GABA is a slightly different type of amino acid, but it's still an amino acid. It's an abbreviation by itself. The next group down here, which is serotonin, dopamine, and norepinephrine, you might have even heard at least two of those before, belong to a class of molecule that's called a biogenic amine. Biogenic amine means that these are molecules that were made in the body from amino acids. So they're not amino acids anymore, but they used to be, or they started as amino acids, and enzymes changed them into different molecules. Serotonin is made from the amino acid tryptophan. Did I talk about the tryptophan turkey myth before Thanksgiving? No. Right.
You heard of that? Anybody ever tell you, oh, you're going to fall asleep after eating turkey? Because that's bullshit. Excuse me. You fall asleep after eating a big turkey dinner because you ate a lot of food and your body just shuts down and allows all the digestive processes to go. The myth comes from the idea that there is tryptophan in turkey, and tryptophan is used to make serotonin, which is the neurotransmitter that's released from certain neurons, regulating sleep and wake behaviors. Okay. The thing is that turkey is not the best source of tryptophan. Okay. There's more tryptophan present in cheddar cheese than in turkey. So if tryptophan levels in food actually did cause people to fall asleep, you would hear more about nacho comas than turkey comas, and you don't. So that's all just an urban legend. There's actually a great Mythbusters episode, if you ever watched that show, where they test this out. So they had several different conditions, eating either a regular high-caloric turkey-based meal or a high-caloric non-turkey-based meal, or a regular calorie level turkey-based meal, or a regular non-turkey-based meal. And then also taking a supplement of tryptophan amino acid, which you could buy at a health food store or something like that. And they tested people on their ability to play whack-a-mole as an example of alertness. And what they found was eating a regular meal, there was no difference in the ability to play whack-a-mole versus when they're obviously fully awake. Eating a low-calorie turkey meal had no effect either. It was exactly the same effect as eating a regular meal. But then high-caloric meals, whether it's turkey-based or not turkey-based, caused whack-a-mole performance to go down. Interestingly, taking the tryptophan supplement also compromised whack-a-mole ability. Not as much as a high-caloric meal, but it did make some significant changes to a person's ability to do that. And so you can go to a health food store and buy a tryptophan supplement pill as an alternative sleep aid instead of taking
Melatonin.
I was going to say Ambien or something like that. If you don't want to take those kinds of things, you can buy tryptophan pills, and it might actually have some sort of effect. If someone were going to do that, I would actually suggest more likely to take melatonin pills, which is a hormone that regulates night-day cycles in your brain. I think that's probably a little bit more efficacious, which just means works better. I think what tryptophan is actually doing is serotonin is used in the digestive tract as a hormone. And so I think that basically induces the coma-like effects that you get from a high caloric meal. But it's not as effective as eating a high caloric meal. So I don't suggest that eating lots of food before you go to sleep is a great way to increase sleep behavior. But that whole thing about turkey on Thanksgiving is based on the relationship of tryptophan to serotonin, but it's not really true. It's an urban myth. Anyways, the serotonin is made from tryptophan, and its abbreviation is a little weird. It's 5-HT, which is actually an abbreviation of its real chemical name, which is 5-hydroxytryptamine. And the tryptamine is referring to the fact that it's made from tryptophan. So the receptors are called 5-HT receptors, and there are multiple types of that again, but we're not going to get into subtypes. They are all going to cause hyperpolarization through indirect effects. So they're metabotropic receptors, and they hyperpolarize the targets. So serotonin is actually very much like glycine and GABA in the fact that all three of these cause inhibitory postsynaptic potentials. And they can be called inhibitory neurotransmitters, which is technically not the most correct way to say it, but you might hear that. The next two types here, dopamine and norepinephrine, are both made from another amino acid called tyrosine. And there's actually a whole cascade of changes that start with tyrosine. There's an enzyme that converts tyrosine into another amino acid called L-DOPA. And then L-DOPA is modified by another enzyme into dopamine. And then dopamine can be modified into another molecule. No, wait, say that again. Dopamine can be modified by another enzyme into, I think it's norepinephrine. And then norepinephrine can be modified by another enzyme into epinephrine. So dopamine and norepinephrine, and then I put in parentheses here that it's related to epinephrine. They are all actually steps along a pathway that starts by enzymatically changing tyrosine. Cells that release dopamine just have the two enzymes that go tyrosine to L-DOPA to dopamine. Okay. And since they don't have the other enzymes, that's what they release. Norepinephrine releasing neurons have an additional enzyme that converts dopamine to norepinephrine. And then there are cells that have another enzyme that convert norepinephrine to epinephrine. Okay. There may be some neurons somewhere in our brain or spinal cord that do that, but more often where we see epinephrine being released is from cells in the adrenal gland. Okay. Now, we usually think of the chemical that's released from the adrenal gland as being called adrenaline. Okay, adrenal in adrenaline. That's a Latin word, or it's derived from Latin parts. That means adrenal gland is the gland that's on top of the kidneys. Ad means on top of, renal means kidney. If we said that in Greek instead, epi means on top of, and nepher is the root that means kidney. So adrenaline and epinephrine are actually the same thing. So in the adrenal gland, there's an enzyme that converts norepinephrine, not epinephrine, and the adrenal gland releases that. The adrenal gland releases a bunch of other stuff, but that's AMP2 material. But since it's being released from that gland, we really consider that a hormone and not a neurotransmitter. But it's related to norepinephrine, which is a neurotransmitter. Sure, there are those. Now, dopamine has two subtypes of receptors called D1 and D2. Actually, it has 5 subtypes of receptors. D3, 4, and 5 are just variations of 1 or 2. And I can never remember which. It might be like D1, D3, and D4 are the same, and D2 and D5 are the same, something like that. But we just classify them as D1 or D2 type receptors. And D1 receptors cause depolarization through indirect effects. They're metabotropic type receptors, and they depolarize the target neuron. D2 receptors cause hyperpolarization through indirect effects. They're also metabotropic receptors, but they hyperpolarize the target cell instead of depolarize. Okay. And we're going to talk about something before the end of the semester, might be next week, might be the week after, that points out why thinking about the two different types of dopamine is important, because dopamine is released into a region in the brain, and the cells in that nucleus have one or the other type of receptor, and so they react differently to dopamine, and it actually is an important part of how that part of the brain works. And we're going to talk about that later on. And then norepinephrine, and for that matter epinephrine, which is really a hormone, have receptors that are called alpha adrenergic or beta adrenergic receptors. And in adrenergic, notice it has adrene, like the adrenal gland. So it's just receptors that bind to adrenaline type molecules. And there's an alpha and a beta type. There's actually like 3 alphas and 2 betas, or 2 alphas and 3 betas. I think it's 3 alphas and 2 betas. But we don't care about the subtypes too much right here. They're just 2 different types. The adrenergic receptors can cause depolarization or hyperpolarization. They're all indirect, meaning metabotropic receptors. But it depends on which subtype we're talking about, which effect it has. I gave the example of the cardiac muscle thing, and muscarinic receptors
will bind to acetylcholine and slow down the rate that cardiac muscle contracts, therefore slowing down the heart rate. There are beta adrenergic receptors on cardiac muscle too, and when norepinephrine binds to that, it causes the cardiac muscle tissue to beat faster and increases heart rate. So actually one of the things this is an example of a thing that we're going to talk about in AP2, that we have two different systems that are both acting on a target like the heart and having opposing effects. So acetylcholine from one system slows down heart rate, and the norepinephrine from another system speeds it up. And that's going to be part of the homeostatic mechanisms that we'll talk about in AP2. Now, I mention alpha and beta specifically here, although I'm lumping them all together as adrenergic receptors when I'm talking about the postsynaptic effect, because you've probably heard of drugs called beta blockers, maybe or maybe not, but they're usually used for cardiac disease or high blood pressure. And beta blockers, as their name implies, block beta receptors, which means norepinephrine doesn't bind to them. So if you block a beta receptor on the heart, that means norepinephrine can't speed up heart rate. So it keeps heart rate lower, which keeps blood pressure lower and keeps the heart from overworking itself. So it's a good type of drug to use for things like hypertension or congestive heart failure, those kinds of things. So you might have heard of that. That's why I happen to mention alpha and beta here, although at this level, we are just going to consider them all adrenergic receptors. Now, you might have noticed as I was talking through this, I skipped over the column here that talks about elimination, partly because it really kind of breaks the receptor postsynaptic effect combination here. But all of the biogenic amines are taken back up into the neurons that release them. And I wanted to mention that specifically about dopamine, because there are drugs that work on the dopamine system in your body because they have an effect on the reuptake protein. And the main drug that does that is cocaine. Now, there's an internet meme out there that you may have seen that has a picture of a chemical structure of serotonin and a picture of the chemical structure of dopamine, and it's labeled something like the only two things that we care about. Because actually, in the nervous system, and this is a gross oversimplification, but in the nervous system, activity of these two molecules is largely related to our pleasure and reward and wake and sleep kind of things. So they're very important molecules in the brain, and they have a lot to do with higher functioning. But cocaine is an addictive drug because it blocks reuptake of dopamine back into the neurons to release it, so dopamine's in the synapse longer, reacting with its particular receptors longer, and the receptors in the reward center, I think, are D1 type receptors. And so it stimulates the reward center more than normal, and therefore it's a pleasurable experience. Now, that internet meme I mentioned is actually a little oversimplification because these are not the only two relevant chemicals in your brain. Other ones have jobs to play too. Nicotine is addictive because there are nicotinic receptors in the brain in the reward center too. So there's more going on, but this is a trip through understanding different types of neurotransmitters. I really want you to pay attention to the postsynaptic effect. What's really important about neurotransmitters and other signaling molecules is the effect they have is entirely dependent on how they interact with the receptor. Okay. So acetylcholine has two possible effects because some of the receptors are depolarizing and some are hyperpolarizing. Dopamine, same thing. Some depolarize, some hyperpolarize. Adrenergic receptors, same thing. Some depolarize, some hyperpolarize. But the amino acids and serotonin all have only one effect because all of their receptors do the same thing. Glutamate always causes depolarization. Glycine, GABA, and serotonin always cause hyperpolarization.
basically in the brain, if you think about depolarization or excitation as an on switch and hyperpolarization or inhibition as an off switch, a lot of the major things going on in the brain are really interplays between glutamate and glycine or GABA. So it's sort of like on, off, on, off, on, off. There's a lot of things going on in your brain that are like that. And I think, I might be completely misquoting the statistic, but I think about 80% of the synapses in your brain, are amino acid receptors, have amino acid receptors. I might have completely misquoted that statistic, but I think it is a really significant portion, which also kind of gets to that internet meme I said about these two. They make up collectively probably less than 15% of the synapses in your brain, but they influence a lot of important parts of your brain. So it's a little oversimplified, but there is something to be said about these two molecules being very important for things that we cognitively enjoy doing in our life, like sleeping or taking drugs or whatever. Now, dopamine is going to be active in any reward center activation. So, the runner's high and that kind of thing is going to be dopamine released too. But don't take drugs, kid. Anyways, so again, this is a modification of the table that's actually in the book. Another thing about this table that's different from the book is in the book I included a section on neuropeptides, which are kind of neurotransmitters, but also kind of do something slightly different. So I left them out of here because in my class I don't deal with neuropeptides, but I put them into the book because other instructors that might be using the book would probably want some reference to neuropeptides. So I put them in the book, but I left them out here in this version of the table because this is specifically for us, and it highlights the fact that you can just ignore the protein, peptide stuff. Someday I'll add peptides to this table because I'm going to get the publishers to switch out this table for the one that's in the book there. But anyways, questions about neurotransmitters or receptors? Okay, so that's the end of the nervous tissue chapter stuff, finally. Okay. Takes a little while to go through all this stuff, and I want to be sure that you understand it because neurophysiology is, at the cellular level especially, is really pretty complex. So I just wanted to wrap that up. So now we are officially moving on to the last part of the semester, which is looking at the nervous system in broader strokes and understanding the physiology of the nervous system more in terms of behavior and the sort of things that we can easily observe. Okay, you can't easily observe electrical activity in neurons. That's kind of tough to explain. But here we'll be talking about things that are not necessarily so simplistic that you can like, oh yeah, I get that perfectly well. But it's a lot easier to talk about examples because they're behaviors that people perform or have seen before. Or in some of the stuff we'll be doing later on, it's also going to be thinking about abnormal behaviors, and we can watch little movies of neurology patients who have been interviewed, and we can see something about how they behave, and it'll tell us something about how the nervous system works. So in the book, I need to update that. In the book here we have, so we just finished the first chapter from this section. Now we're moving on to this stuff. So we're going to concentrate on the anatomy of the nervous system chapter. And for the most part, the part 1, 2, 3, and 4 assignments in this folder are from that chapter. But this chapter obviously is called Anatomy of, so it's mostly about the anatomy. The physiology of the nervous system, again, at more of the behavioral level, is distributed across the next 2 chapters. So there's one chapter looking at the somatic nervous system, which is, as I describe it, conscious perception and voluntary control of our bodies. And then another chapter about the autonomic system, which is homeostasis for the most part. We will be concentrating on homeostasis in AMP2, so we will not get to this chapter. Actually, in AMP2, we're also going to look at sensory systems, which is the conscious perception part. So part of this chapter on the somatic nervous system, we're actually going to hold off to look at until next semester. So a little bit of this chapter will be relevant to what we're doing now. So I will pull a couple of things out of this chapter as I'm talking about how the nervous system works. And then I'm also going to go to this last chapter here in the center column, the neurological exam. And this is where we'll think about things like abnormal behaviors. The neurological exam is a clinical assessment tool. It's not a test that you're going to be taking. I have to say that explicitly because in the past, people have said, when are we going to be taking that test called the neurological exam? It's not a test, it's not a graded thing, it's a clinical tool. Okay. But by looking at specific neurological functions in people that have brain injury or something like that, we can get a really good idea of how the brain is responsible for certain things. I've said it a few times before, you're taking anatomy and physiology together because structure and function go together so intimately. So by looking at the loss of function, somebody can't do something that we can see in the neurological exam, we can then figure out what part of the brain, what structure in the brain is affected and therefore responsible for that function in normal people, normally behaving people. For example, if you ask somebody to stick their tongue out, stick out your tongue and say, ah. Part of doing that in the doctor's office is to look in the back of the throat for signs of infection or something like that. But also, little known to most people that have to do this for the doctor, it's also testing your ability to stick your tongue out. Okay, so you should stick your tongue out straight like that. But if there's damage in a specific part of the brain, you can't stick your tongue out directly. So if somebody sticks their tongue out when they're just asked to do that and they think they're sticking it out straight, it might go off to one side. Okay. And the nice thing about the neurological exam and that particular example of that tool to test it, the tongue sticking out to one side or the other tells you which side of the brain has the damage. Okay. So if your tongue goes off that side, that means that the left side of your brain has some damage, specifically the brainstem. The reason for that is that the muscles that are in your tongue are controlled in a lateral fashion, meaning that the left side of your tongue is controlled by nerves that come from the left brainstem. The right side of your tongue is controlled by nerves that come from the right brainstem. I think I said that right. And so one of the weirdest examples of muscle contraction is the tongue, because we usually think of contraction as shortening muscles and making things move inward towards our body. But when muscles in the tongue contract, you stick your tongue out. Okay. So when you stick your tongue out and it goes off to one side, that's because the side that it's pointing away from is contracting properly and pushing out. And the side that it's bending towards, those muscles aren't contracting. And so it's limp, if you will, on the bad side, and the good side sticks out and sort of pushes into the limp side a little bit. I'm not going to try to explain tongue motion in a lot of detail, but if you ask somebody to stick their tongue out and it goes off center, it's because the side that it's pointing to has a problem. And we can assess that kind of thing from the neurological exam. But we can also look at things like the ability to remember lists of information, or the ability to use language in various ways, or to walk a straight line, or to touch your nose with your fingertip. Those kinds of things are all looking at behaviors or neurological functions that are affected by specific parts of the brain. Okay? And so this is a great way to see how function and structure are tied together. Because you can say, oh, they're not doing that right, and you know exactly where in the brain to look for a problem.
So that's what we're going to kind of cover. We've already sort of started talking about this chapter on the anatomy of the nervous system. The first section is on the embryology, and I did that in lab, so I'm not going to repeat that in here. And then it gets to talking about the central nervous system, and I believe I used some of these pictures in lab last time to show us around the cerebral cortex. I'll go into a little bit more detail. We didn't look at this picture, for example, or this picture. And then we'll go into just briefly explaining some things about other parts of the brain. In lab, the anatomy that we're looking at of the brain is really about the cerebral cortex. So in lecture, we're going to talk about the deeper parts of the cerebrum and the diencephalon, brainstem, and cerebellum a bit more. So we'll go through this stuff. And then we'll talk about the spinal cord, and actually we'll also talk about the spinal cord in lab. Depends on time, how much time I'm going to spend talking about the spinal cord in lab and how much I'm going to spend talking about in lecture. Then we're going to talk about circulation in the nervous system, central nervous system, which is going to look more closely at what the blood-brain barrier is about and the substance called cerebrospinal fluid. So we'll get to that. And then very, very briefly in lecture, I'm going to address some things in the peripheral nervous system, but really I'm going to address what I want to out of this mostly in lab, where we'll talk about, okay, so first off, we've really already talked about ganglia because we looked at unipolar neurons in that slide. It's about as much as I'm going to go into ganglia. And then for the nerves, we're going to look, well, we've already sort of looked at the structure of nerves in a couple of slides. But otherwise, we're going to think about nerves as they are broken down into cranial nerves or spinal nerves. And that'll bring us through that part, which will wrap up that chapter. In the somatic nervous system chapter, there's really only 3 parts here. We're going to deal with the sensory perception part in AMP2. And so there's a little bit about central processing, a little bit about motor control, which is really about the spinal cord. I just happen to have it covered in the somatic nervous system chapter of the book, but I'm going to use that stuff to supplement when I talk about the spinal cord. So that's essentially what we're going to get into. Now, I think it's probably a good idea to hold off on starting talking about that stuff until, well, I guess it'll be next week, next class. So we'll wrap up here for the day. I'm sure you're terribly upset that you're missing out on 12 minutes worth of me talking next week, but I think it's just a nice split to make. So we finish off the nervous tissue stuff, and then we'll start the nervous system stuff next time. Finish out the semester over the next, what is that? So 3 more classes and 2 more labs. Okay, so that's what I sort of ran through is sort of what we're going to do for all of those 5 meetings. Otherwise, have a nice weekend, and